Recording in progress.
Assalamualaikum warahmatullahi wabarakatuh and good morning everyone. Today the lecture is about the formation of germ disk and growth of the embryo. It is actually the continuation of the previous lecture I gave you on Monday. So last week was the first week and this week will be the second, third until 8 weeks. All right, before we go into the lecture of this week, this is what we already learned during the last lecture, which was related to the conception to week 2, which is also known as pre-embryonic period, where during this period, the fertilized ovum undergoes mitosis, formation of morula, appearance of blastocyst, and the implantation of the blastocyst. All right, today we're going to look into the embryonic period. Well, this period is from the 2nd to 8th week. The germ layers formation from bilaminar germ, three germinal germ layers, the placenta development, and main body system formation. Okay, this is a very quick revision on the first week. What happened during the first week? Okay, number 1 is fertilization. Fertilization that produces zygote. Okay, in all this process, the different phases that are present in the fertilization, what need to be done to the sperm before the process of fertilization, and what are the important products of fertilization. Let's go through again if you can answer my question. Now we go, what happened after the fertilization is the cleavage of the zygote, which produces the blastocyst. Okay, what is the blastocyst? But what is blastomere? What is morula? What is blastocyst? And which of these product will be implanted into the uterus wall? I hope you know which one. Okay. And then the implantation of the blastocyst. Okay, I'll give you the answer. Okay, the blastocyst will be implanted into the endometrial wall, the inner wall of the uterus. What happens if it is implanted outside the uterus? It will become ectopic pregnancy. Okay, this lecture carry many learning objectives. Okay, it's not only 4. Okay, let's go one by one. Okay, this lecture you will be learning what are the germ disks, how many germ disks are present. Okay, and the formation and components of the germ disk, development of amniotic cavity and fetal membranes, primary germ layers and extra-embryonic structures. This is what you have to know, the gastrulation, and how the 3 germ layers are established by the primitive streak. Okay, you have to know this. Okay, and development of some primitive structure and their fate, formation of 3 germ layers and their derivative, and formation of intra-embryonic xenon and body cavities, and a bit about cephalocaudal and lateral folding of the embryo, and neurulation and its significance, the notochord formation of limb buds, and just these of major bone structure that begin to form during the embryonic periods. Okay, there are 12 objectives in this quite heavy lecture. Okay, let's go slowly. Okay, the second week of development. Okay, remember what happened during the last lecture? It was implantation. So implantation is established at about 6 to 7 weeks after fertilization. Okay, what happened after implantation? Okay, the inner embryoblasts start to differentiate into the flattened bilaminar embryonic discs. Okay, there are 2 bilaminar two layers of the embryonic disc, the epiblast and the hypoblast. You may look at my pointer, okay? The epiblast and the hypoblast. Okay, the epiblast is the upper, thicker single layer cells, and then the lower, thinner single layer cell is called the hypoblast. Epiblast blue, hypoblast yellow. And what happened next? Second small cavity appears in the blastocyst between the epiblast and the cytotrophoblast. This cavity is known as the amniotic cavity. Surrounding the amniotic cavity is the amnion or amniotic membrane. Okay. And this is our blastocystic cavity. Okay, this cavity now become the exocoelomic cavity. They are now called exocoelomic cavity, which is surrounded by the exocoelomic membrane. Okay, I hope you understand one so far. Okay, this picture showing the bilaminar. This is the bilaminar germ disc, the epiblast, the taller cell layer on top of hypoblast. Hypoblast is the flattened cell layer. And the amniotic cavity is the cavity that is located between the epiblast and the cytotrophoblast. The amniotic cavity is surrounded by the amnion or amniotic membrane. Okay. There is also remember our blastocystic cavity, become exocoelomic cavity, and the exocoelomic cavity is lined by the exocoelomic membrane, right? Exocoelomic membrane that lining the exocoelomic cavity. Okay, the next day. What happened the next day? Exocoelomic cavity membrane are modified to become the primary yolk cell. So they are now primary yolk cells. How to differentiate between the exocoelomic cavity and the primary yolk cell? Okay, you can see in this picture, there is an additional layer that forms outside the exocoelomic cavity. This layer is known as the extraembryonic mesoderm. Extraembryonic mesoderm. Okay, there are a layer of loose connective tissue that forms between the cytotrophoblast and the primary yolk sac layer. They are known as extraembryonic mesoderm. This extraembryonic mesoderm is later forming the chorionic sac. At the same time, you can remember that since trophoblast, the tissue of trophoblast that is fused, that has no cell membrane, that start forming small cavity within it. This cavity is known as lacunae. And these cavities are known as lacunae. Later, the lacunae will fill with mixture of cellular debris from uterine glands. Uterine gland is present in the maternal endometrial wall, and maternal blood from eroded endometrial capillaries also in the endometrial wall of the uterus. This lacuna is really, really important. They provide nutrient to the blastocyst, to the growing the embryo through diffusion. So what happened during second week so far? So it's the primary yolk sac formation and formation of extraembryonic mesoderm, which later developed into chorionic sac, and formation of lacunae. lacunae that present within the syncytial trophoblast that provide nutrient to the blastocyst. Again, another picture showing the same thing. Okay, extraembryonic mesoderm, the primary yolk sac. This is the bilaminal embryonic disc, and this is the amniotic cavity and the amnion or amniotic membrane. Okay, this is the syncytial trophoblast that contain multiple lacunae that contain marginal blood from eroded capillaries in the uterine wall. Okay, so by this time, you already learned what is the bilaminal disc and how is the development of the amniotic cavity happen, and the primary yolk sac. Okay, 10 to 12 days after fertilization, implantation is now fully complete. Okay, during the rest of second week of development, the lacunae fuse. They fuse to form a lacunal networks. The cavity developed within the embryonic mesoderm. Remember the embryonic mesoderm, the loose connective tissue between the primary yolk sac and the cytotrophoblast. The now forming cavity is known as extraembryonic coelom or chorionic cavity. Extraembryonic coelom or chorionic cavity continue to enlarge, makin besar, and eventually they fuse and pinching the primary yolk sac. So from this size, they pinch the primary yolk sac into two part of the yolk sac. The upper one become the secondary yolk sac, and the lower one, the smaller one, the minor one, become exocoelomic cyst. The primary yolk sac then detach from the embryo and disintegrate into collection of the exocoelomic cyst, which ultimately degenerate. Later, they can disappear. Okay. From the cavity of the exocyst, embryonic coelom or chorionic cavity, they enlarge, they fuse, they pinch out this very big yolk sac and dividing the yolk sac into secondary yolk sac, and the primary yolk sac become exocoelomic cyst, and later it will disappear. Okay. After the formation of the chorionic cavity, the extraembryonic mesoderm becomes split into two layers. Okay, one is the structure that lining the secondary yolk sac, the part that is lined by the green colored line. This layer is known as extraembryonic splanchnic mesoderm. And the yellow colored or the yellow orange colored line is known as extraembryonic somatic ataupun somatoclinic mesoderm. Somatic and splanchnic. Okay. Secondary yolk sacs, the extraembryonic coelom, the extraembryonic membrane became two part that lining the secondary yolk sac is known as the splanchnic layer of extraembryonic mesoderm, and the layer that is lining the cytotrophoblast and the amnion is known as the somatic layer of extraembryonic mesoderm. So what happened so far? So at this stage, the primary yolk sac is already developed, And what happened to the primary yolk sac and secondary yolk sac? How is this evolution take place? Primary to secondary yolk sac. What is extraembryonic splanchnic mesoderm, and what is extraembryonic splanchnic mesoderm? Right. Now, move on to the chorion and gestational sac. Chorion is formed by the extraembryonic mesoderm. Remember the extraembryonic somatic mesoderm, the layer that is lining the cytotrophoblast and amnion, plus 2 layers of trophoblast. Okay, so it contains the extraembryonic somatic mesoderm, okay, plus the cytotrophoblast and the syncytial trophoblast. The chorion forms the wall of the chorionic sac. So this is the chorionic sac. This is the chorionic sac. Okay. It is also known as gestational sac, within which the embryo, this is the embryo, and the secondary yolk sac, and the amnion cavity, amnion membrane, is suspended by a structure called the connecting stalk. Okay, this is ultrasound scan showing the gestational sac. Okay, this is a very advanced ultrasound scan where the doctor can actually you can actually see the yolk sac, the fetal pole, and amniotic cavities up here within a very well gestational sac. Okay, this is the gestational sac. This gestational sac is also known as chorion. Okay, another picture showing this is the not so advanced ultrasound scan. The scan can only locate the gestational sac, the developing embryo, the yolk sac, and also the amnion is not really visible in this type of scan. But there is a gestational sac. So it's confirmed that this lady is pregnant after a positive urine pregnancy test. Okay, now go to the chorionic villi. What are those chorionic villi? Villi mean villus. Villus mean finger-like structure or projection. Okay, the primary chorionic villus. This is secondary chorionic villus, and this is tertiary chorionic villus. What are the difference between these three? The primary chorionic villus is a projection of finger-like structure that contain the core of the cytotrophoblast. The core of the villus is only made of the different numbers of cytotrophoblast. Secondary chorionic villi. In secondary chorionic villi, these cytotrophoblast is no longer being in the middle of this villi. They are now replaced by the mesenchyme. Mesenchyme is the tissue of the extraembryonic mesoderm. Okay, and the tertiary chorionic villus contain the blood. This is fetal blood vessels. They are now known as tertiary villus. What is actually this chorionic villi? Later, they will form this important structure which is known as placenta. So you have another lecture regarding placenta later. Okay, you can see here the chorionic villi. This is the fetal portion of the placenta. This is the maternal portion of the placenta. Okay, up to this hour, minutes, you learn the chorionic sac and chorionic villi. What are these two structures? And now we go to the third week of development. Okay, the third week means 3 lamina. Third week, 3 lamina. So during the third week, the bilaminar embryonic disc converted into 3 germ layers of primary germ layers. Primary germ layers is the 3 germ layers through a process called gastrulation. Gastrulation. The primary germ layers consist of ectoderm, mesoderm, and endoderm. Okay, how is the gastrulation process take place? Okay, it begins with the formation of primitive streak. Okay. Do you know what is streak? Streak is just a line, primitive line. This line appear about 12 to 14 days after fertilization. Where is it? At the epiblast part of the embryonic disc. Remember our embryonic disc contain epiblast, the taller layer cells, and hypoblasts, the flatter layer cells. Okay, this midline streak starts appearing from the caudal end of the embryonic disc at the cloacal membrane and slowly ascend up towards the cranial end. Okay, from the caudal end to cranial end. As the streak elongates, it forms the primitive nodes. Primitive nodes, and in the middle of this primitive node, there is a primitive pit, which stops growing halfway up. So the primitive streak is developed until grow until up to point. A narrow groove forms behind the extending streak, which is known as the primitive groove. Okay, very complicated process, but I think it's easy to understand. Okay, another picture showing the same gastrulation process. So the gastrulation begin with the formation of primitive streak. Okay, the epiblast of the bilaminar germ disc. This midline streak start appearing from the caudal end up towards the cranial end. Okay, where is the cranial end? The cranial end is up here, and this is the caudal end. In echo, in the right? They slowly ascend up towards the cranial. As the streak elongates, the primitive nodes. The primitive nodes is um, the, the expanding part of primitive streak. The primitive streak, it has its cranial end or expanded, which is known as primitive node. And in the middle of primitive node, there is primitive pit. And these primitive streak grow up until this point only. Okay, only halfway of the disc. And at the same time, the narrow groove forms behind the extending streak called the primitive groove. So the primitive groove. Make sure you know how to describe this gastrulation process and how is what the component involved in the primitive streak, primitive nodes, and primitive pits. Okay, again, another picture showing the same thing. Okay, the midline streak start appearing from the caudal, slowly ascend towards the cranial end. As the streak elongates, it's form a primitive node at its cranial end nodes. Okay, primitive pits is located in the middle of the primitive nodes. It stop growing halfway up there in the disc. And the primitive groove is formed behind the developing primitive streak. So this is the process of gastrulation that we can describe externally. What is actually happening behind this process? The streak emerges due to the proliferation and migration of cells from the epiblast to the hypoblast. So they proliferate. Not only they proliferate, they also migrate from the epiblast here to the hypoblast. Because of this migration, they form a new layer in between the original epiblast and hypoblast, which is known as the intraembryonic mesoderm, the mesoblast. Some of cells of epiblast come to lie in hypoblast layer and displace its cells so that the hypoblast eventually is replaced by a new layer of cells, which is known as the endoderm. While the epiblast, they become the ectoderm. So this is the primary germ layer, which contain the ectoderm, mesoderm, and endoderm. Okay, ectoderm, mesoderm, and endoderm. Okay, these are the derivative of the primary germ layer. For ectoderm, they form epidermis, the nervous tissue in nervous system, and retina, which are amongst the other structure that form from there. Ectoderm. The mesoderm forms the cardiovascular system, the skeletal, the renal system, the red blood cells, and also the smooth muscles. While the endoderm forms the gastrointestinal tract, the respiratory tract, the bladder, the and the organ that is related to the gastrointestinal tract. What happened to the primitive streak? Okay, on day 16, the primitive streak spans about half of the length of the embryo. So that means it reached its maximum length. As gastrulation process, gastrulation means the formation of the three germ layers process, the primitive streak gradually becomes shorter and shorter. By day 22, the primitive streak represents about 10 to 20% of the embryo length, and it is completely disappeared at about day 26 after fertilization. So, what have we learned? We already learned the gastrulation. What is the definition of gastrulation? You have to know the process and product of gastrulation. And it's nice way to know the fact of primitive streak. Now we go to the notochord formation. Okay, notochord formation. This is the continuation from the gastrulation process. Okay, some cells from a primitive node, some cells from the primitive node. Remember our primitive node? Okay. That grow only halfway of the embryonic disc. The primitive node and pits migrate cranially to the precordial plate. So yeah, migrate cranially towards the precordial plate. The precordial plate. These mesenchymal cells migrate within the new mesenchyme layer between the ectoderm and endoderm, forming a midline cord called the notochord, and this process is called the notochord process. Processing cord, which is known as notochord process. Notochord is a cellular rod running along the longitudinal axis of the trilaminar embryonic disc. It extends from primitive nodes to precordial plate. Precordial plate. Where's the precordial plate located at? It is near the oropharyngeal membrane at the cranial end of the bilaminar germ disc. These indicate the future site for the vertebral bodies. This cord soon developed a lumen. and is then termed the notochordal canal. So this is the notochordal process. The canal developed within the notochordal process is known as notochordal canal. This cord extends halfway along the length of the disc from the primitive node to the oropharyngeal membrane with the cranial line. This is the oropharyngeal membrane. This is the notochordal canal. Vertebral bodies form around the notochord. So this is notochord. Vertebral bodies form around the notochord later in its development. Once the vertebral column has been formed, the notochord degenerates. Notochord degenerates. Once the vertebral column formed completely, the notochord within the vertebra degenerate, except at some part of the intervertebral disc. The disc that is located between two vertebrae is permanently there, which is later it is called as nucleus pulposus. Nucleus pulposus is the remnant of notochord. Have you understand? Notochord also has another function. It induces the ectoderm covering it to increase in thickness. This ectoderm that is thick, then the other part of the ectoderm is known as neural plate, the beginning of the central nervous system. The process of CNS development is called the neurulation. So notochord is also important in inducing the ectoderm thickening for the neurulation, formation of central nervous system. Neurulation is the term for the formation of the nervous system. It is the formation of the neural tube as notochord develops upperlying ectoderm thickness to form elongated plate of thickened epithelial cells called the neural plate. Remember the neural plate? The neural plate elongates as far as oropharyngeal membrane. The neural plate invaginates along its central axis. Invaginate maksudnya ke dalam. Along its central axis to form a longitudinal groove. So, form a longitudinal groove. On each side of the neural groove, it is called the neural form. The neural groove, this is the neural form. Neural form move closer and closer each day and fuse with one another, converting the neural plate into the neural tube. Neural tube soon separates from the surface ectoderm. So it's forming the neural tube with its central canal, which is called the neural canal, and the neural crest. You can see the neural crest is from this the neural plate. Initially, neural tube has two openings. One is at its cranial end, which is the cranial neuropole, and one is at its caudal or tail, which is known as caudal neuropole, which communicates with amniotic cavity. The neuration is complete when both neuropoles closed. The cranial neuropole closed at day 25, while the caudal neuropole closed at day 27, 2 days later than the cranial. Neural crest, okay, remember the blue structure part of the neural plate in the previous slide. It's called the neural crest. Neural crest is a special population of cells arises along the lateral margin of the left neural folds. Okay, During neurulation, the cells detach from the neural plate and migrate to many specific locations in the body where they differentiate into a variety of structure. So these are all the neural derivative: bone of the facing skull, ganglia of cranial, spinal, and automatic nerve, C cells of the thyroid gland, adrenal medulla, Schwann cells, and glial cells, arachnoid and pia mater, and melanocytes. So we learned that notochord formation, the process and the products of notochord formation, and the neurulation process. Now we go to the last, the growth of the embryo, from 3rd to 8th week. Okay, this is formation of embryo during the 3rd week to the 8th week, where the trilaminar germ is begin to assume human embryonic form due to encephalocaudal folding and lateral folding. Okay, actually, this is a video. Well, I will show this video. I will upload this video in on Elip along with this lecture later on. Okay. The folding of the embryo is a significant event that transforms the flat embryonic disc into the somewhat cylindrical structure. So this folding, cephalocaudal folding and lateral folding, occurs simultaneously, same time. So now the flat embryo is become the cylindrical structure embryo. Okay, this is the videos. I will upload it on Indeed. I don't know why I can't actually play this video. And a little bit about formation of intra-embryonic xylem. So remember, this is the intermediate mesoderm, which is the new layer that formed between the ectoderm and the endoderm. Intra-embryonic xylem is a large cavity developed within the lateral plate mesoderm. During the 2nd month, the intra-embryonic xylem is divided into 3 body cavities, which is the pericardial cavity, pleural cavity, and peritoneal cavity. And formation of somites. Somites are these are all the somites. Okay, somites are block of mesoderm located on each side of the developing neural tube. Somites develop from segmentation of paraxial mesoderm, which is from the somatomias. Somites form distinct surface elevation on the embryo. First somite appear at the end of 3rd week, which is at day 20. Okay, this is neural tube, and this is somite. Paraxial mesoderm. Paraxial mesoderm begin forming blocks called somite in neck. And spread in cranial sequence. There are 42 to 44 pairs of somites at the end of 5th week. Well, you don't have to memorize all this. But there are 4 occipital, 8 cervical, 2 thoracic, 5 lumbar, 5 sacral, and 8 to 10 coccygeal somites. A somite contains sclerotome, and dermatome that separates into dermatome and myotome later. Okay, sclerotome will form vertebra and intervertebral disc, which is the skeleton, and the dermatome become the dermis and the skin, and the myotome from the skeleton. These are the importance of somites. Somites give rise to most of the axial skeleton. Bones, cartilage of vertebral column, ribs, sternum, part of the occipital bone. And also voluntary muscle of the neck, body wall, and limbs, and part of the dermis of the neck and trunk. The number of somites can be used as one of the criteria for determining the age of the embryo in early stage of development, which is only for research purpose, not in clinical practice. Okay, you already learned the cavalicoid folding, lateral folding, somite formation, and components of somites. Okay, now the very last part of this lecture, the formation of limb buds. At a 6-week-old embryo, the terminal portion of the limb buds become flattened, and form the hand plate and foot plates. Hand plate and foot plates. They will then separate from proximal segment by the circular constriction. Okay. And later, the second constriction divides the proximal portion into two segments. Okay. And the main part of the extremities can be recognized. This segment and this segment. Right? This of major body structure developed during embryonic period. Number one is central nervous system, the brain and spinal cord. The head is ear, nose, mouth, and eyes. Primitive gastrointestinal tube and respiratory organ, which form during embryonic folding. Cardiovascular system is the earliest to function. Limb buds develop at 5 or 6 weeks. By the 8th week of development, the embryo has all the basic organs and parts except those of the sex organ, which develop somewhat later than 8 weeks, up until 12 weeks. Okay, I think that's all. Again, this is actually another video for you to watch. 6 weeks of human development in 1 minute. So, no worries, I will touch on it later. I think that's all. I wish you all the best. And thank you for attending to my lecture. So this lecture can actually be, I don't know. Okay, I think that's all. Bye bye.